Hey everyone, I'm Giovane and this is Physio Webinars 2022. Thank you very much for being here today. This is an event organized by graduating students from the Physiology graduated program of the University of São Paulo, Institute of Biociences from the University of São Paulo, USP. This is our team. We organized 3 amazing talks that happened during November and December with speakers from different countries talking about distinct topics on physiology and their intersection with other areas. Please follow us to get more information. Now it's time to get to know more about today's speaker. Professor Dr. Gary Hardiman joined the Faculty of Medicine, Health and Life Sciences and Global Food Security Institute at Queen's University Belfast, also known as QUB, in 2018 as Chair of Food Systems Biology. His laboratory at QUB operates in the field of systems biology, the object of which is the study of biological systems, including genes, RNAs, proteins, metabolites, and cells in a focused manner, and organs, organisms, and populations in a broader context. The ongoing research focus areas are studying the effects of microplastics on marine and human health, prostate cancer in the context of racial differences and nutritional deficiency, examine the impacts of long-term space travel, develop a model of opioid abuse in rats, toolkit optimization for better integration of omics datasets into genotype-phenotype predictions, and computational analysis to interpret the development of metabolic diseases mediated by no-coding RNAs. That's a lot. Gary. Hardiman is the co-founder and CTO of Northern Ireland-based precision medicine company Atomics Datamation Limited. So, Dr. Hardiman, thank you very much for your presence here today. After the talk, you have up to 30 minutes to answer the questions that are on the chat. Everyone can ask questions in the YouTube chat. They can be in Portuguese, English, and Spanish, and our team will select and translate if that's the case, and send the question here to us. And this event will be recorded and will be available in YouTube after the transmission. Without further ado, I will give the word to you now, and we'll be here in the background in case you need something. Now you can share your screen. Thank you very much, Gary.
Okay, lovely. Okay, lovely. Just double check that you can see my screen in presentation mode. Yes?
Yeah, it's perfect.
Okay.
Yeah.
Perfect. Okay, wonderful. Good afternoon, everybody. Thank you, Giovane, for the introduction. It's a real pleasure to be here today to give this sort of talk at Physio Webinars. So greetings from a very damp, wet, cold sort of Belfast December afternoon. So the title of my talk is SpaceOmics, Aging and Prostate Cancer, a Systems Biology Perspective. So I'm going to start the talk by, taking us through the concept of bioinformatics, like basically, sort of what is bioinformatics. And I really like the definition that Russ Altman, a professor of bioengineering genetics medicine and sort of biomedical data science at Stanford University sort of has put in place for this. He defines bioinformatics as when he builds a method, when I build a method, I design it to have a certain performance characteristics. I build it using best engineering practices. I validate that it performs as I intended. I create it to solve not a single problem, but a class of similar problems that all should be solvable with the software. And then I write papers about the method, and these are engineering papers. And for me, this is a really beautiful definition of bioinformatics. Computational biology then, because the two are often compared, Russ Altman defines it as when I use my method or those of others to answer a biological question, I'm doing science. I'm learning new biology. The papers that result report new biological knowledge. These are science papers, and this is very much computational biology. So bioinformatics and computational biology, two things that my laboratory does. And one of the things that we've seen over the last 15 years or so is this paradigm shift in how, as biologists, we're able to, analyze data and sort of deal with data. If you did a PhD 30 years ago, you were likely looking or working on sort of one gene or one mRNA at a time, or you were characterizing one protein or one, small region of the genome, looking maybe at methylation patterns in that region of the genome. What ultra high throughput sequencing has been, sort of able to do for us is it's been able to take a lot of assays that were in place for analyzing sort of DNA and RNA and sort of DNA bound to protein. And it allows us to effectively, look at it in its entirety. So we can look at entire transcriptomes, we can look at entire methylomes, we can look at how transcription factors and how sort of various proteins, histones and so forth, interact with DNA. And this is very important because this allows us to study regulation. And this paradigm shift that we've seen in high throughput technologies, probably advanced most by genomics, but we've seen parallel advances in mass spectrometry and various other high throughput technologies, has allowed us to do what we call systems biology. And systems biology is based on the understanding that the whole is greater than the sum of the parts. It's responsible for some of the most important developments in the science of human health and environmental sustainability. And it's very much a holistic approach to decipher the complexity of biological systems that starts from the understanding that the networks that form the whole of living organisms are more than the sum of their parts. And I sort of got that very nice definition from the Institute for Systems Biology. So I moved to QUB from the United States about a little over 4 years ago and have set up effectively what is a systems biology laboratory. And we work on different things. And you might ask the question, you've got a lot going on there. You're not focused on one particular research area. But my answer to that typically is that we work on data. We're particularly interested in looking at the effects of man-made contaminants, particularly in the context of food safety and food security, and how these contaminants impact marine and human health. We're interested in prostate cancer, and I'm going to talk a little bit about that later today in the context of racial differences and nutritional deficiency. An area of research that my lab got into over the last 4 or 5 years was space travel, and looking at the impacts of things like nutrition and mitigating some of the adverse effects of space travel on human physiology. With American and US colleagues, we're developing a rat model of opioid abuse to better understand the biological basis for addiction. We're developing sort of toolkits to integrate omics data into genotype, phenotype predictions. I'm going to talk a little bit about that today. And we're interested in looking at non-coding RNAs in the context of cancer and metabolic diseases. So when you sort of think about sort of biology, you sort of have to think about it in the context of time. So we have sort of temporal scales, but also when we're dealing with biology, we also have to look at spatial scales. So we can start with macromolecules, that sort of will make up organelles in cells, and then cells come together to form tissues, tissues become organs, organs comprise an organism. And, we're interested in studying, all of these different levels, but ultimately one of the things that we try to do is to study, what's going on in biology at the population level. And that might be in the context of ecosystem biology, or it might be in the context of, epidemiological studies of populations. So we're interested in spatial scales, but we're also interested in temporal scales, because what happens is that, biological, events can occur in sort of micro and milliseconds, things like enzyme catalysis and metabolism. Gene regulation will happen sort of on the order of seconds to minutes, hormonal regulation sort of on the order of sort of hours, and then development and aging, you know, sort of over the context of years and decades. So one of the things that we sort of normally have to look at is, you know, sort of how we sort of deal with this type of data. And the fact as well that, you know, sort of if you have a change in gene expression, that may not be manifested at the protein level, you know, until sometime after the change at the protein level. So two students, recent graduates of the Masters in Bioinformatics program, Masters in Computational Genomics and Bioinformatics, I should say, here at QUB, sort of wrote this very, very nice sort of review article that we just published in iScience, basically sort of looking at multi-scale modeling and a sort of promise for spaceflight biology. Spaceflight is associated with a lot of hazards, including radiation, exposure, and microgravity. And systems biology approaches have the potential to provide new insights into space biomedicine. So I'm going to, you know, just go off on that track here for a few minutes. Again, I've mentioned the context of sort of, you know, temporal and sort of spatial aspects that we need to be aware of in terms of, you know, travel in the solar system, you know, to get from here to you know, the International Space Station, or to get to the moon is typically, so you know, anywhere from 28 hours to 3 days. But the big interest at the moment, I think, is in going to Mars. And we're hearing a lot about this, you know, in the media. Mars is one of the few places in our solar system where life similar to Earth, life effectively, may be able to survive. So this makes it a location of special concern for human exploration. And it's estimated that depending on sort of favorable planetary alignments between Earth and Mars, that a planned mission to the red planet would involve a 400-day transit there and back, and potentially a 500-day stay. So this type of Earth-dependent deep space mission presents many, many challenges for astronauts, because you effectively have to feed astronauts. So they need nutrition. And, you know, this isn't easily doable. You know, for example, the International Space Station in low Earth orbit, it's, you know, it's possible to sort of provide, you know, and replenish sort of nutritional supplies. But once you sort of move away and sort of start getting you know, further away from Earth, this gets harder to do. So Thomas Cahill, a PhD student working with me here in Belfast, this has been sort of the sort of topic of his PhD, nutritional challenges and sort of countermeasures for space travel. We published this in Nutrition Bulletin about 3 years ago. So there's technical challenges associated with providing enough food with sufficient nutrient content, bioavailability, and increased shelf life. An issue is that environmental stressors that you find in the space environment will impact physiology and in turn will affect nutritional status. Increased exposure of radiation, which we see in the space environment, causes changes in the gut and liver, sort of two key organisms that might facilitate nutritional deficiency. And so current or potential countermeasures for these challenges are being explored, including the use of bioregenerative systems such as growing crop or biomass, or introducing torpor, which is this reduced metabolic state to sort of reduce nutritional needs. So my lab has been involved in a few collaborative efforts, one with the European Space Agency. I'm looking forward to traveling to Madrid next week to meet some of these colleagues, where we've been sort of involved in revamping sort of SpaceOmics in Europe. And then when I was in the USA, I was sort of part of the NASA sort of Gene Labs consortium, and Dr. William Da Silveira, who was a postdoc with me in the US and in Belfast, and now who has his own laboratory in England, in Staffordshire, led on this paper that was published in Cell a couple of years ago, which was titled A Comprehensive Multiomics Analysis reveals mitochondrial stress as a central biological hub for spaceflight impact. And essentially, that was a large collaborative paper that involved many, many different groups at NASA and, across the world. And the study basically looked at assessing the impacts of spaceflight at a molecular level on cells and tissues. And this involved, looking at rodents and sort of rodent organs, sort of cell lines from humans, including fibroblasts, T cells, endothelial cells, and hair follicles, and essentially asking the question, sort of what happens at an omics level to these sort of, cells and tissues, organs and organisms in space. And, the types of analysis that were done were very broad, things like DNA methylation analysis, expression analysis using RNA-seq, sort of protein analysis, metabolite analysis, and, interesting bioinformatics pathway and sort of modeling analysis at the back end of this. And what all of this sort of integrative analysis of these multi-omics data sets showed was that there was a significant enrichment for mitochondrial processes, as well as innate immunity, chronic inflammation, cell cycle, circadian rhythm, and olfactory functions. So at the center of a lot of these physiological problems that astronauts experience in space was a deficiency in sort of, or a decline in mitochondrial function. And one of the nice things about this particular study was that, you're very familiar, I'm sure, with these two astronauts, Mark and Scott Kelly. Scott Kelly spent a year up on the International Space Station, or close to a year on the International Space Station, and Mark Kelly, his brother, also, both of them are retired astronauts at this point, but they were, a fantastic resource, to have two identical twin brothers, one that remained on Earth, one that was up in space. And, the ability to be able to sort of, confirm some of the findings, that we had sort of uncovered in the study in sort of these individuals. And what we saw basically was that in sort of Scott Kelly, there was evidence of altered mitochondrial function and DNA damage, which was found in the urine and blood metabolic data, which would indicate sort of mitochondrial stress as a consistent phenotype of spaceflight. So the systems biology analysis that was done as part of this study pointed at mitochondria, and then this was sort of validated in sort of this astronaut twin study. So I'm not really going to go into much more detail about this, other than I think it's a great paper, well worth a read. What my lab has sort of been doing is looking at ways of potentially sort of mitigating sort of some of this damage, if you wish, that occurs in space. So torpor is a state of decreased physiological activity that you find in an animal. It's usually involves reducing body temperature and reducing metabolic rate. And this enables animals to survive periods of reduced food availability. So we see this in bats, particularly this time of the year, particularly here in Northern Ireland where it's cold and damp. And we see it as well in bears. So basically, these animals sort of hibernate to go into this sort of state of torpor, the state of decreased physiological activity. So the idea of using suspended animation or induced torpor has been, proposed as far back as the 1960s, consistent with, the very first space voyages of man to the moon. And it's currently being explored by the European Space Agency. So the idea is that the approach would involve crew sedation, along with reduction of body temperature, lowered metabolism, and would consequently lower oxygen and nutrient requirements. Nutrition and hydration could be administered intravenously in a solution known as total parenteral nutrition, or TPN. So my lab, in the context of a lot of toxicology work looking at the impacts of plastics and various other endocrine disruptors, had been using the zebrafish model. We've used zebrafish model for quite some time. And I like zebrafish because it has many advantages. It has high fecundity, short generation cycles. It's relatively cheap to maintain compared to rodent models. There's a very good build of the zebrafish genome available for bioinformatics work. The genome is easily modified. The embryos in adults are transparent. The embryos develop externally, and nearly all of the genetic code is expressed or active during early life stages. The most interesting thing, though, I think about zebrafish is that 70% of protein coding human genes are related to zebrafish genes. And when you think in the context of disease, this number actually increases to 84%. So therefore, I've always argued, and others have, I think, that the zebrafish model is a really, really good proxy for human health assessment. So when I was still in the US, in Charleston, South Carolina, at the Medical University of South Carolina, about 5 or 6 years ago, myself and my colleagues, Dr. William Da Silveira and Dr. Ludovine Renaud, they were two postdocs that were working in my laboratory at the time. We sort of submitted a grant, a small sort of pilot feasibility study to NASA through the EPSCoR program that South Carolina is involved in. And the idea was that we proposed developing a zebrafish model of induced torpor and radiation exposure. So zebrafish are poikilothermic. They vary their body temperature based on their ambient temperature by modulating their gene expression and metabolism. Their ambient or optimal temperature is 28.5 Celsius. And, the literature had shown that you could actually sort of, reduce temperature over a period of time. And that sort of the zebrafish, in reducing its body temperature would also reduce its metabolic rate. So there was a precedence for sort of using zebrafish based on the published literature. We were interested in zebrafish because they have modulatory neurotransmitters in their brain that are similar to humans. And we decided that we would use the hormone melatonin to promote a sleep-like state and reduce locomotor activity in this organism. Melatonin is a potent antioxidant, and it's a regulator of the circadian rhythm. And we used it to increase sedation and reduce activity. So we were successful in sort of getting this pilot grant, and then this allowed us to do this experiment, where we set this experiment up, where we had groups of fish where they were maintained at 28 degrees Celsius, the ambient temperature, without sort of being exposed to melatonin. And we had a group where, over a period of 4 weeks, we reduced their body temperature from 28 Celsius to 18 Celsius. So we reduced the temperature by 2.5 degrees Celsius per week. So this was gradual to prevent sort of, stress and, cold shock to the zebrafish. And then what we did was we mimicked the exposure that sort of humans would get on a journey to Mars and back. So we had sort of 2 days. We had a 4-week acclimation period, and then on day 2, the fish were exposed to radiation via radiation flare, and then they were exposed again on day 8. So basically, sort of, we had a control group at ambient temperature, we had a reduced temperature group, we had a reduced temperature group that was exposed to the drug, melatonin, and then we had a group that were exposed to radiation. So the radiation that we opted to select was low-dose radiation, 0.3 grays, again, similar to what would be experienced on a 6-month journey to Mars, to allow assessment of the potential protective properties of torpor. So we had a control group, like I said, melatonin, temperature, torpor, radiation, torpor, and radiation. We teamed up with one of my colleagues who was at MUSC at the time, Dr. Shireen Chan, a zebrafish biologist, and she and I had collaborated on some other behavioral analysis on zebrafish. And we use this instrument, which is a Danio Vision, which allows you to track motion of zebrafish. So it allows you to monitor zebrafish activity. So the activity is a measure of, sort of how much they move. Are they sort of in this resting state, or are they quite active? And what we saw effectively was that reduced temperature alone, or in combination with melatonin, significantly reduced the activity levels in zebrafish compared to the ambient 28.5 Celsius control. Melatonin alone, though, without the low temperature, did not significantly lower the activity score. So it required both sort of the colder temperature and the melatonin. So this basically validated the torpor-like sort of model in zebrafish, showing that it's possible to increase the resting phase, exploiting the combination of melatonin treatment and reduced temperature. So this happened as I was sort of moving to Belfast, and Dr. William De Silveira came with me to Belfast. And then I was able to recruit Thomas Cahill, who was a PhD student, who then started to dive in and start to sort of analyze this model. And Dr. Hao Wang, a former postdoc, a PhD student and postdoc of mine, was also involved in this work. So what effectively we did was, we were well aware of the fact that ionizing radiation is one of the major obstacles with space travel, because it generates oxidative stress, and this can damage cellular structures such as DNA. It leads to things like genomic instability, telomere shortening, extracellular matrix remodeling, and persistent inflammation. So the first tissue in this zebrafish model that we decided to look at was the gastrointestinal tract, the GIT, as this can lead to leaky gut syndrome, perforations of the gut, motility issues, which would impact the gastrointestinal tract functionality. And in a space environment, it would sort of completely, impact and affect its nutritional status. So what Tom did was he dove in. He started to analyze RNA sequencing data that we had generated from the GIT, the gastrointestinal tract, of these sort of different conditions that we had available for the model. And so interestingly, one of the first things we saw was that when we looked at which genes were differentially expressed from a comparison of the sort of torpor and the control group, we saw that the torpor group experienced a downregulation of key subunits of the electron transport chain proteins such as NADH dehydrogenase, succinate dehydrogenase, and cytochrome oxidase. And this sort of, was consistent with reduced energy production. The other thing that we saw was that there was a perturbation of circadian rhythm pathway genes compared to the low-dose radiation. Perturbation of circadian rhythm pathway genes to low-dose radiation compared to the control group. So this then is a biological pathway topological diagram, and it shows the position and regulation of differentially expressed circadian rhythm genes. And some of the genes that were sort of impacted in terms of their expression were upregulation of PER and REV ERBA, and the downregulation then of the genes CRY-CLOCK, BMAL, and aren't-like in comparison to the control group. So this would suggest that low-dose radiation actually impacts and perturbs circadian rhythm. Another finding was that the protein digestion and absorption pathway showed an upregulation between the ambient control 28.5 radiation versus 28.5 control, and it pointed at peptidases and amino acid transporters in response to the low dose radiation exposure. So again, here we're seeing protein digestion and absorption pathway being impacted by radiation exposure. We also saw that there was a perturbation of genes encoding proteins involved in endoplasmic reticulum associated protein degradation. what we found was that there was an increase in expression of chaperones involved in detecting misfolded proteins, as well as those involved in the ER degradation pathway. What we saw was that there was an increase in anti-apoptotic genes and a decrease in pro-apoptotic genes. We teamed up then with my colleague, Dr. Ian Overton at the Patrick Johnson Cancer Centre here in Belfast to do sort of a network analysis. We use this program called Net and C analysis. And what this showed us basically, when we sort of generated networks sort of from these different groups, was that each condition showed, substantial overlap with the other conditions, but there were some noticeable differences. Those being the upregulation of genes in the torpor and radiation group that conferred radioresistance and sort of promoted survival. So essentially, we think that this is kind of an interesting model that we're sort of further developing because induced torpor, if applicable in a spaceflight situation, would lead to an estimated reduction of 88% of total consumables. It would also lead to a decrease in the need for power systems, life support, space vehicle size, and the need for consumable storage space. And this would reduce fuel needs and overall mission cost. So to sort of reiterate, low-dose radiation, where the zebrafish were kept at ambient temperatures while exposed to the radiation, they experienced elevated reactive oxygen species, increased stress signaling, and halting of cell cycle. Whereas when we induced torpor, and the zebrafish were exposed to low-dose radiation, what we saw was that the fish experienced a reduced metabolic rate, lower oxygen concentration, so therefore reduced harmful effects from reactive oxygen species, increased pro-survival cell signaling, and increased protein maintenance. So I'm going to switch gears just a little bit now and sort of move from sort of space into the concept of aging. But the two biological sort of phenomenons are actually related, because what we tend to see up in space is we tend to see accelerated aging of cells and of tissues. So the whole area of aging is, again, something that a lot of researchers that are involved in space biology are also studying because of the overlap between the two phenomena. So senescent cells, are interesting because they provide an emerging target to study diseases of aging. So there was quite a nice review published in Nature Reviews in Drug Discovery by Childs et al. Back in 2017. And they show that chronological age represents the single greatest risk factor for human disease. One explanation for this is that the mechanisms that drive aging might also promote the disease that we tend to see in older age. Cell senescence, which is a permanent state of cell cycle arrest, which is induced by cellular stress, has recently emerged as a fundamental aging mechanism that also contributes to diseases of later life. So things like cancer, atherosclerosis, and osteoarthritis. And therapeutic strategies that safely interfere with the detrimental effects of cellular senescence are gaining significant attention, with several programs now sort of nearing human clinical studies. So what we were interested in my laboratory was, prostate cells, because sort of prostate cancer research is something that I've been quite interested in, for the last decade or so. And we were interested in understanding how microRNA and long non-coding RNA expression networks modulate cell cycle and DNA repair inhibition in prostate cells when they senesce. And to go back to, what I was saying at the start in terms of, kind of software development, we suddenly found ourselves pivoting from being a biological research lab to needing to develop software to be able to handle some of the data that we wanted to look at. One of the things that we were interested in doing was taking social media concepts and applying these to the non-coding RNAs, the microRNAs and the long non-coding RNAs, to try and develop sort of new networks, looking at the effects of microRNAs on mRNA expression or long non-coding RNAs on mRNA expression. So what we did was we looked at sort of two key concepts that you find in social medias. One is the concept of centrality, and this can highlight which microRNAs are more important than others in a specific biological system. So from a social media perspective, I think that Elon Musk is sort of a perfect example of this at the moment, because he is very, very, very central to, if you take Twitter as an example, he has, many, many millions of people who sort of follow him. So he's very much an influencer. He's very much, someone that's sort of central, to a network. Many of us then, like on this call, probably follow Elon Musk on Twitter. And if we follow Elon Musk on Twitter, we're probably likely to follow, maybe some other, tech giant, or, someone that sort of, if you're interested in Elon Musk, it's likely that you're interested in, several other people that you will follow on Twitter or on social media. So many of us, will have similarity, I think, in terms of our interest in social media. And this concept then is the concept of similarity. And what you can do with both centrality and similarity is that you can sort of define these sort of using edges and nodes. And centrality is a measure of the degree. It's the number of edges that are connected to a vertex. And, like I said, it can highlight which microRNAs are more important than others in a specific biological system. Similarity then, basically, if two vertices in a network are structurally equivalent, if they share many of the same network neighbors, that would indicate similarity. So basically, we didn't kind of set out to, develop a software tool, but we were interested in this compound, bisphenol A, and its impacts on sort of zebrafish on both microRNAs and mRNAs. And we found ourselves developing a tool sort of based on these concepts to look at, how microRNAs, these non-coding RNAs, regulate mRNA expression. So the premise behind the tool is that the microRNA acts via downregulation of its mRNA targets. So this is the canonical function of microRNAs. And the regulatory effect of microRNAs is dependent on the cellular context. So basically, we assumed the canonical function of the microRNA. Cellular context was important because it had to be able to regulate the mRNA of interest in the tissue of interest. And microRNAs that regulated the greatest number of targets had a greater impact on the phenotype. So this was the concept of network centrality. And then the list of common targets of two microRNAs in that context can be used to infer how similar they are, network similarity. So what we were able to do then was we were able to dissect differentially expressed microRNAs, and we're also able to dissect, the interaction between microRNAs and mRNAs, and sort of, put this into sort of functional groups. And we published this in Genes back in 2018. So more recently then, what we did was we looked at prostate epithelial cell senescence, where we got prostate epithelial cells from a company called Planetics that are in San Diego. And these were cells that had been derived from a healthy 53-year-old male with no history of prostate disease. And we took the cells through 7 population doublings until they senesced. And then we sequenced mRNA and microRNA from senescent and active cells. And the library approach that was used for sequencing was one that also allowed us to look at the long non-coding RNAs. So what we did was we extracted long non-coding RNAs and their target genes from the list of differentially expressed RNA transcripts. We used the long non-coding RNA heterogeneous regulatory network integrator, which is Longhorn. This basically sort of derived from some work done with the Cancer Genome Atlas prostate adenocarcinoma data set. So basically TCGA-PRAD basically provides us with the list of long non-coding RNAs that regulate mRNAs in the prostate. So we built sort of miRNA target networks using an OR package called Multimere. And then we used our tool MirMapper to identify the most central miRNAs in the biological network. And this, I think, is more interesting because often what happens, when scientists will analyze sort of microRNA data is that they will pick the one or two that have the greatest fold change in expression levels and then make the argument that these are probably most important in driving the biology. Whereas what we were interested in doing was we were interested in taking microRNAs and long non-coding RNAs and asking the questions as to which of these had the most impact in terms of network centrality. So we were able to sort of, rank microRNAs and rank long non-coding RNAs, and these are the top 10 of both in terms of their influence on biological networks. So what we did was we then modeled all of this data. I don't expect you to see this. It's really small. I'm just going to summarize, what we saw, like when we made this network representation of long non-coding RNAs and microRNAs, when we compared senescence versus sort of active cells, were that there were a series of gene targets that basically belong to the prostate cancer KEG pathway that sort of were differentially expressed and were quite important and central to this network. One in particular was EGFR. Basically, this gene target is highlighted. In this figure here, we also highlighted FOXM1 and CDK1 from the cellular senescence pathway, and we effectively built out networks of these genes. So effectively, what we were able to do was we were now able to, using this tool, we were able to describe the central role of non-coding RNAs in the senescence process of primary prostate cells. And we showed that senescence induced the perturbation of regulatory non-coding RNA gene networks, which led to the downregulation of several genes that were involved in cell cycle and DNA repair processes, which are two of the key hallmarks associated with cellular aging. So our analysis put long non-coding RNAs at the center of this deregulation, and we described some functions that had not been previously reported or studied. And this opened up the possibilities for the development of new biomarkers and therapeutics for the treatment of aging and cancer. I'm now going to kind of switch from sort of cell lines into sort of humans, again, going back to my idea of kind of spatial organization in biology. I've been involved in some NIH-funded work in the US, where, along with collaborators, in Charleston and South Carolina, we have been, developing systems approaches to interrogate gene expression patterns in African American men presenting with clinically localized prostate cancer. So here, in this picture, you've got two men. They're probably, I would guess, late 50s, maybe early 60s. The man on the right is Caucasian, he's white. The man on the left is African American, he's black. They're friends, they play football. Both may develop prostate cancer, but the man on the left, the black man, the African American man, he's more likely to have sort of more aggressive prostate cancer. And I'm going to talk about that. if we kind of look globally at sort of top cancers per country for mortality in 2018, we can see that lung is the predominant cancer, but it's followed quite closely by prostate cancer. As I mentioned, there are significant health disparities in prostate cancer. There are racial disparities in prostate cancer outcomes. So prostate cancer will disproportionately affect Black men of African ancestry in terms of their incidence, morbidity, and mortality, even after adjustment for stage of the disease. African American men have a 2 to 3 times greater or increased risk of developing prostate cancer and have a much higher mortality rate compared to white men. They also have higher levels of this biomarker for prostate cancer, prostate-specific antigen, or PSA, and they have higher grade tumors than sort of European American men when presenting at the same stage of the disease. So in terms of genomics research, ultimately, what we want to do with genomics and multi-omics research is to improve precision medicine approaches. And the success of precision medicine and sort of genomics ultimately means recruiting people from underrepresented populations for genetic studies. And this is a challenge because there's a lack of diversity in genetic studies to date. One of the challenges, for example, is that African-American men remain grossly underrepresented in prostate cancer genomic studies. They account for 12% or less in the TCGA, the Cancer Genome Atlas database, and less than 3% of therapeutic trial participants. So what we wanted to do, I think, was we wanted to see if we could link stress and health disparities in prostate cancer. And there's an emerging hypothesis about racial disparities in cancer risk and outcomes, is that psychological and social stressors impact cellular stress responses. But there's very little empirical data available on racial differences in cellular stress responses among men who are at risk for adverse prostate cancer outcomes. So what we wanted to do was we wanted to try and interrogate the prostate transcriptome with the single biopsy core and examine molecular differences in gene expression patterns relevant to the disparities that exist in sort of prostate tissue between African American and European American men. So we focused on a cohort of men who had presented either with early stage prostate cancer or were at risk for prostate cancer. And we carried out a series of genome-wide expression profiling experiments using RNA-seq. So we published this last year in Cancers, MDPI Cancers, and sort of this, like I said, was a large collaborative effort between my lab, Steve Savage's lab, Shanita Hughes-Halbert, sort of now at USC, and Sebastiano Gattoni-Celli, and sort of Beth Wolf. Again, it was a big study with a lot of different people, which I think is a theme, I think, in my talk today, that sort of systems biology-based research really is not done in isolation. It's done with collaborative teams such as these. So what we did was we, were able to generate very high-quality sequencing data. Initially, we looked at 60 patients. We had 27 European American, we had 33 African American, and we generated RNA sequencing data from the prostate biopsies from these individuals. And we, did fairly deep sequencing. We, sequenced to a depth of at least, 60 million reads per prostate biopsy sample. And we generated about 3.8 billion reads total across all patients. One of the nice things, because we were working in a clinical setting, was that we had, all of the clinical characteristics associated with all of these patients. For example, things like the number of positive cores, prostate-specific antigen levels, HbA1c, which is a measure of diabetes, because one of the problems with prostate cancer is that it sort of has comorbidities associated with it. We also had measurements on things like vitamin D, which I haven't spoken about in this talk, but that has been the topic of other sort of research in the context of prostate cancer that we've done. Typically, low vitamin D levels or deficiency in vitamin D tends to link with worse sort of prostate outcomes. So lots of interesting clinical data, lots of interesting omics data. Rebecca Morgan, a PhD student working with me here in Belfast, was involved in the analysis of this data. And, one of the things often that, that you sort of have to try and keep reviewers happy with in this context is, that you have enough power in your study to be able to do this, because a lot of work that goes on in the context of cancer, and particularly prostate cancer, has been based around GWAS studies with thousands of patients. I mean, you come in with sort of a smaller cohort of 60 patients, you have to be able to sort of prove that you've got enough power in your analysis. So I think we were able to do this. We provided power calculations based on the number of replicas that we had, the depth of the sequencing, a coefficient of variation 0.4, which is based on sort of an outbred population like humans. And also we used a program called DSeq2, which is one of my favorite pieces of software. We were able to, find genes that were upregulated or downregulated in African Americans relative to European Americans. So one of the things we saw initially was that there was an interesting sort of signature. 3,224 genes were significantly differentially expressed in the prostate when you compared European American and African American men. When we asked the question, where did these genes map to? It was refreshing to see that they mapped with a co-expression signature called genes upregulated in prostate cancer samples from sort of previous studies. And when we started to look at sort of biological pathways, we saw some of the key pathways that were involved in cancer. So PI3 AKT signaling, which plays an important role in the development and maintenance of castration-resistant prostate cancer, a lethal form of the disease. This was one of the key pathways that came up. We also sort of saw a neuroactive ligand receptor interaction. This is sort of an important pathway which comprises G protein coupled receptors, ion channels, and ligands, which function in modulating neural plasticity, memory processes, and behavior. We also saw ECM receptor interaction, the extracellular matrix, which is involved in the maintenance of cell and tissue structure and function. Interestingly enough, one of the things that did not come in the pathway analysis, but came up in sort of functional enrichment studies, was that we saw a lot of signatures associated with the ribosome RNA binding, ribosomal RNA binding. And one of the interesting things with this is that this is indicative of a basic molecular stress response. So this would suggest that the African American sort of prostate biopsies had a stress-based signature that was differentially expressed relative to the European Americans. And this basic molecular stress response has been linked, sort of with very, very primitive organisms like, bacteria when they're stressed. Structural constituent of the ribosome was one of the sort of key terms that came up, and alterations in sort of ribosomal biogenesis are one of the things that we tend to see in malignant transformation and progression. We also, like I said, saw RNA binding proteins being sort of differentially expressed. So effectively what we did was we whittled this down to a non-redundant signature of 187 genes that were differentially expressed between sort of the black men and the white men. And we made a network. And one of the things we saw when we looked at this in network space was that there was a high degree of interactivity amongst these 187 genes. So clearly this then would suggest that there was some interesting or important biology underlying this. When we looked at the 187 gene signature across all of our patients, we saw that there were differences in gene expression patterns, which wasn't surprising because this was how we had extracted these. But what we saw was that there was a gradation, basically, where, there were, sort of extremal sort of signatures, at sort of both ends of the spectrum. And in particular, there were 5 patients that had elevated expression of most of these genes, or elevated downregulation of sort of some of these genes. And when we went back and examined the clinical characteristics of these patients, we noted that they were sort of worse off from a health viewpoint. They had elevated Gleason scores, greater numbers of positive core biopsies, higher triglyceride levels, and lower C-reactive proteins. And of course, it didn't surprise me that they had diminished serum vitamin D levels. So we teamed up with my colleague, Dr. Beth Wolf at MUSC. She ran something called a sparse principal component analysis, where we were interested in trying to dissect out, some of these attributes, if you wish. And what we saw was that was strongly associated with these 5 patients, very, very significantly, with these patients showing the largest value for this component. So our conclusions here basically were that what we wanted to do was identify the systemic milieu in which prostate cancer develops and potentially progresses, especially as it relates to a comparison of European and African-American men. We interrogated the prostate biopsy transcriptome. We analyzed, it for the very first time, and compared gene expression differences relative to prostate cancer between European and African-American men presenting with early stage prostate cancer. Comparison across racial groupings uncovered differences in transcriptomic profiles, and this indicated a differential stress signature between African-American and European Americans, which allowed separation of patients with more clinical characteristics. So this takes me to my last slide. Like I said, systems biology is very, very much collaborative research, and there are people that I definitely need to acknowledge. I'd like to acknowledge my colleague, Dr. Starr Hazard, who has since retired. He was a long-term colleague of mine at MUSC. I would like to acknowledge Dr. William da Silveira, a Brazilian scientist who worked with me in the US and here in Belfast, and now has his own lab at Staffordshire in England. Ludovine Renaud, who was a postdoc who worked with me in Charleston. And then I would like to acknowledge my group here in Belfast, particularly Rebecca Morgan, who's been involved in some of the prostate cancer work, and Thomas Cahill over here, who has been involved in the sort of torpor study. I'd also like to thank Beth Wolf, Shireen Chan, Tucker Williamson, Dongjun Chung, a collaborator of mine at Ohio State University, Ian Overton here at the Patrick G. Johnson Center for Cancer Research. And lastly, I'd like to sort of thank my sort of funders. None of this research is possible without funding. So I'd like to thank the NIH, NSF, and NASA. And with that concludes my talk. So I am happy to take questions. So back to my host.
All right. Thank you very much for the talk. It was fascinating. SpaceOmics as a whole and the transposition of social network concept to RNA analysis is just, I don't know, it's so creative. It's really fascinating. Thank you very much for the presentation. Now we have some questions. The first question is from Maiara Moreira. She asks is the mitochondrial damage an essential response of the ambient difference? Other than radiation, does nutrition and gravity play an important role in this intracellular damage?
Yeah, so that's a really good question. So mitochondrial damage is something that we see, you know, in the context of space travel, and it's a combination of, you know, the harsh environment in space. So it's a mixture, really. You can't really take these things in isolation. You know, I mean, up in space, you have exposure to radiation, and you have microgravity, which is really, really harsh on the human body, because we weren't designed to exist in an environment of microgravity. Mitochondrial damage is also something that is an issue with you know, aging as well. You know, as we age, you tend to see mitochondrial damage. So one of the things actually that we're sort of starting to do here at Queen's at the moment.
So I think your camera just froze for a moment. We'll wait for. Oh, so your transmission is kind of. You said the first thing you done, and we lost the connection.
Sorry,
Okay.
I'll repeat that. Yeah, it's
okay.
It's getting later in the evening here in Belfast. Everyone's on Netflix, I guess. Lower bandwidth. Yeah, so we're starting. So there's two things. One is, so mitochondrial damage is a central tenet of you know, the damage that we see in aging and up in space. So how does one mitigate that? So you can mitigate that with pharmaceuticals. You know, like you can look at things like resveratrol would be one, for example, you know, which is a good anti-aging compound. One of the other things that we're interested in is something called coenzyme Q, which is a nutritional supplement. And one of the other things that we're interested in is this Chinese herb called daylily. So this is sort of something that we've sort of gotten into recently, because we work within an Institute of Global Food Security. So if we can develop nutraceuticals, you know, kind of, if we can mitigate this sort of using nutritional approaches, this is interesting. So what I was saying was that Dr. Colin McClure, who's a colleague of mine here at Queen's, who's a Drosophila biologist, he and I are kind of starting to explore this concept where we're replicating what we did with zebrafish in the fly. It's a little bit easier because, you know, the fly life cycle, or, you know, lifespan is even shorter than the zebrafish. So yeah.
So thank you. And we also have a question from Thaysa. She asks, humans are endotherms and homeotherms. We do have a change in our body temperature normally, but in general terms, we don't go into torpor. What would this torpor would affect human physiology? So yeah. I know there's a lot of things to talk about that, but
yeah, you know, what can you yeah, hang on, yeah, some questions. Okay, so to answer that, yes, how would you replicate this in humans is the key question here. And I think that you know, the answer to that brings in, you know, kind of studies on hibernations that are happening, you know, like animals that hibernate. So that's one angle that we're definitely exploring. So one of the great things about systems biology, and one of the great things about comparative genomics, is the conservation that exists. You know, so we can take data from bears, we can take data from bats, we can take sort of data from space-flown animals, and we can sort of look at sort of overlaps between, you know, hibernating animals and sort of, you know, sort of transcriptomic changes that actually sort of happen up in space. And this is something that we're sort of actively doing. We have a paper, you know, almost accepted, you know, where we've sort of gone in and we've sort of done that with the liver. And so that's one approach, is that you can sort of, you know, use knowledge from you know, sort of biology and sort of comparative genomics. The other, of course, and we're doing this as well, we've teamed up with a colleague of mine, sort of who's an anesthesiologist, Dr. Deacon Farrell. He's based in Los Angeles, and he's very much interested in this because, like myself, he's interested in space biology, but he's also interested in, the concept of, how, anesthesia sort of can be applied to sort of tackle this in a space environment. And it raises a lot of issues because, like, how are you going to administer anesthesia up in space? Is one crew member going to need to be, active to do this? And there are, cycles of, rotation where, sort of one astronaut is awake and the others are asleep, and they switch this out. It also would have to be pretty simple, I think. Simple probably isn't the right word, but not complex, I think, in terms of how the anesthesia would be delivered. And then, I think what we're seeing is that, and this is, from conversations I've had recently with Deacon, is that there aren't that many, anesthetics. There are a few anesthetics that are used over and over again. And the question is, will any of these be applicable in a space environment? So he and I are sort of working on a review article in the space at the moment. So stay tuned. I think we're going to hopefully get that out early next year.
I guess no one is planning to go to space on the next year or 2025. So I guess we're good for now.
Yeah, we're good. Yeah, there's definitely some time. Yes. Yeah.
Yeah. Maybe vacation in other places. So
Right.
we have our third question is from Amanda Guerreiro, and she asks, what can we expect about the effects of low-dose radiation and torpor-like states in circadian rhythms? I just want to add something to that because I'm a chronobiologist myself, and we know that there's a temperature compensation for circadian rhythms. It's really robust, but I didn't know that low dose of radiation can also affect that, because I always thought that for circadian biology, the main effect of space travel would be the extraterrestrial environmental cycles, but the change in the molecular clockwork for this radiation makes things a lot more complex. So you,
yeah, I think at the level of human biology, I haven't been in space. I'm not likely to ever be in space. But I think what's interesting, I think, is that one of the challenges that astronauts have in space is poor sleep quality. And a lot of that, I think, is because the regular circadian sort of day-night rhythm that we have is perturbed and upset. And I think what we saw, in our zebrafish model, that sort of these signatures were actually there and, front and center. So the question then again comes back to, is this something that could be mitigated against, therapeutically, or preferably sort of like using sort of targeted nutrition? And I think this is something that, my group is interested in, and a lot of my collaborators are interested in as well.
That's really nice. I guess we have one more question on YouTube. Let's see. Okay. Amanda Guerreiro also asks if you believe that due to reduced access to proper healthcare and economics, African Americans present more incidence of prostate cancer. So I'll just copy that because my accent in English is kind of horrible.
I got that. That is a very relevant question. It's a very important area of research that my collaborators, particularly Shanita Hughes-Halbert in the US, sort of works in that space. One of the challenges, I think, that we saw, particularly in South Carolina, when we were trying to develop any research programs and trying to do outreach into the community, was that there was a certain distrust of medical practitioners and of scientists by a lot of people. And I think that, for example, the Tuskegee sort of experiment, kind of in the US, that sort of famous, experiment where, prisoners were sort of exposed to syphilis, and kind of weren't told, and this was done over a sort of a period of time. The damage that has done is sort of still realized today. There is a lot of sort of mistrust of medicine, of science, which kind of really is unfortunate. And that kind of really opens up an entire can of worms, really. Even, I mean, in terms of, I think, the backlash that a lot of scientists got in the US during the COVID pandemic, whereas those of us that are trained as scientists, data is presented to us, we look at it rationally and we sort of make a decision. But literally, that got turned upside down over the last few years. So without getting off point, when you're dealing with a population group that's economically disadvantaged, and when there's a history there of that population group not being sort of treated very, very well by medical practitioners, there's a lot of work that needs to be done by sort of scientists that sort of want to involve these patients in their studies and sort of want to be able to sort of help and sort of create benefit. So one of the things I guess I myself didn't appreciate was, until I moved to South Carolina, the whole area of sort of disparities research, because to me it seemed kind of like obvious. It's like you obviously need to help people from sort of kind of poorer and sort of more disadvantaged backgrounds. I really hadn't been exposed to that, but it really is sort of a fascinating area of science, the whole area of sort of disparities research. And, it's something that is very, very active at MUSC in South Carolina. And I still collaborate with a lot of colleagues there that are quite active in that space.
Surely. And Amanda also asked, I lost the last bit of the question. She asked if the sunlight that is going into my face, so is the sunlight also a factor for developing prostate cancer? Because you were talking about ethnic groups, and maybe skin color has an impact on that.
Yes, absolutely. So I didn't talk about that study today. So back in 2018, we did a study where we looked at vitamin D levels. And what we did was we looked at not prostate biopsy, where prostate cancer is starting. We actually looked at prostatectomy tissue. So basically, the cancer had advanced to a point in these men where the recommendation was that the prostate be removed, because if it wasn't, like, these were still primary cancers, but if the prostate wasn't removed, the potential for, kind of metastatic cancer was great. So the standard of care then in that case is to remove the prostate. So we had a cohort, not a large cohort, but I think we had 27 patients, where we had a mixture of African American and we had a mixture of Caucasian. Most of the African American patients were deficient in vitamin D, and it's for two reasons. One is, the darker your skin color is, the harder it is to make vitamin D from the sun. Okay, that's the first issue. The second issue, though, is that unless you're a construction worker and you're outside sort of, all day long in blazing heat, it's very, very hard to make enough vitamin D from the sun. You have to get vitamin D, through your diet, And so what we were seeing with a lot of the African American patients was that the diet was poor. Like, basically it was sort of poor diet, which was linking with sort of poor low vitamin D levels. And I think, as we all know, vitamin D is, I would probably argue, the most important hormone in the, in the body, because if your vitamin D levels are deficient, you are at risk, for many, adverse disease outcomes, cancer just being one of them. Like low vitamin D tends to associate as well with elevated inflammatory levels. So, so what we saw in that study was that it was a supplementation study where some of the patients were actually given vitamin D for a period of 3 months. So post prostatectomy, actually, I'm sorry, it would have been after biopsy and before prostatectomy. So there was a 3-month window where the patients were given a regimen of either vitamin D or they were given a placebo. And those that got the vitamin D, the vitamin D levels had sort of come up to be comparable to people that were sufficient in vitamin D. But the interesting thing, though, was that the prostate, the signatures of inflammation in the prostate had gone down. They'd been reduced. So vitamin D looked like it was anti-inflammatory in that regard. Yeah.
So that's really a question that the system biology, like this integrative view of things, is really the way to address the question, because you have to, because I was not thinking directly on vitamin D. I was thinking mostly about skin cancer and maybe metastasis, but it's true, the whole system works as one, and we have to think in everything at the same time. So it was really nice to have you here today, Dr. Hardiman. Thank you very much for the talk and accepting our invitation. I'm sure everybody is looking forward to hear more about your research lines. They're so interesting. And I remember to everyone that this presentation will be recorded here on YouTube, because we're having graduate issues today, so people can check it later. And thank you for everyone who participates as a spectator, as someone who makes questions, as a speaker. And thank you, everyone. We hope to see you in 2023, and stay tuned. So see you in 2023.
Okay, thank you.
So I guess we're no longer live. So